All right, everybody, welcome to our class tonight. I'm going to just take us through a couple announcements. I know I'm letting a couple of you in. I've already started the recording. For this Sunday, you have your first lecture quiz due, and this lecture quiz will be done using Proctorio. It's worth 10 points. And some of the questions on that quiz will be things like knowing the different type of bonds, so understanding hydrogen, ionic, covalent bonds. You might see like a matching question with those, understanding the different parts of the atom, the electron, the proton, the neutron, understanding homeostasis and how our bodies regulate homeostasis using a receptor, a control center, an effector. So that gives you a good start on some of the questions for that first lecture quiz. And that'll be due this Sunday at midnight, so September 12th. And then the only other thing you guys have to do this Sunday is our second lab, which is an introduction to food macromolecules. And a lot of these macromolecules will be covered in this kind of second part of the biochemistry chapter that we'll go through today. Again, with these Lapster labs, you have an unlimited amount of time to do them, and an unlimited number of attempts to do them as well. And it always saves your best score. So feel free to work through those. Tonight, then, we're going to do part 2 of chapter 2, which is the basics of chemistry. And I'm going to go ahead and jump right into it. Part 2, we kind of are halfway through this PowerPoint. Part 2, we'll talk about biochemistry. And again, as I'm lecturing, I don't give you guys study guides for exams, but I try to point out important things you should know. I won't guarantee I'll cover everything, but that's kind of how I help you guys out. You won't need to know, so someone asked in the chat if you need to know how to identify the bonds by the pictures. No, all you need to know is kind of descriptions of like what is a covalent, ionic, a hydrogen bond. So yeah, there will be no picture questions regarding bonds. Good question. All right, we'll let a couple more people in. And if you missed the first couple minutes, you can always watch the recording later. I gave you some hints about what to do for the lecture quiz that'll be due this Sunday. But I'm going to get started here right with biochemistry. I'm going to try to be done in 45 minutes tonight. So we talked about the basics of chemistry in the first part of chapter 2, and now we're going to dive into biochemistry. So we'll talk about some of the molecules specifically that make up living things, and specifically the human body. So biochemistry is the study of chemical composition and the reactions of all living matter. So we're no longer talking about your desk or the glass of water that you're drinking. That's non-living. Now we're talking about living matter. So we call this biochemistry. All chemicals are either organic or inorganic. And we might see those words organic and inorganic pop up again throughout the semester. Organic compounds are water, salts, and many acids and bases, or inorganic compounds are water, salts, and many acids and bases that do not contain carbon. And what makes an organic compound usually does contain carbon. So these will be all of our carbohydrates, our fats, our proteins, and nucleic acids that most often contain carbon. They're usually large and they're all covalently bonded. So they all share electrons. Both inorganic and organic compounds are essential for life, that we'll see. Water is the most abundant inorganic compound. It makes up about 60 to 80% of the volume of our cells. And it's a very unique inorganic compound because of its very interesting properties. So it has a high heat capacity, meaning that you have to get water extremely hot before it will boil. It has a high heat of vaporization that has polar solvent properties, meaning it gives water the ability to dissolve substances very easily. It's very reactive and it provides kind of a cushioning mechanism to it in the body. The high heat capacity, so that it kind of goes through all of these characteristics of water. This is the ability to absorb and release heat with little temperature change. So this gives water the ability to prevent sudden changes in temperature. The high heat of vaporization, meaning evaporation requires large amounts of heat, so it's a useful cooling mechanism. And the polar solvent properties just means that it can dissolve and dissociate ionic substances like table salt. So it can dissolve salt, glucose, Water can form hydration layers around large charged molecules like proteins, and water is our body's main transport medium that we'll see. So here is just showing how water can easily dissociate solutes like salt in water. So the ability of water, it can kind of surround the different ions. So it'll kind of break apart the sodium from the chloride. Water kind of surrounds those ions and it's able to dissolve, things like that. Water is reactive. It's a necessary part of certain reactions. And it's cushioning, so it protects certain organs from physical trauma. So for example, cerebrospinal fluid is made up mostly of water, and that helps to cushion your nervous system organs. So we have cerebrospinal fluid kind of surrounding and within the brain that help as a cushion. Salts are another ionic compound that's inorganic. It will dissociate into separate ions in water, and it will separate into a cation, which is a positively charged molecule, and an anion, a negatively charged, but it will not include the hydrogen and hydroxide ions, which will be separate. I won't ask you really any questions about salts. All ions, which are salts, are called electrolytes because they can conduct electrical currents in a solution. So that's the idea of where an electrolyte comes from. Ions play a very specialized role in body functions like sodium, potassium, calcium, iron. Those are some of the main important ions in the body that we were going to talk about a lot. And then these are some common salts in the body. And again, I don't ask you too many questions about salts. Acids and basids are electrolytes that will ionize and dissociate in water. So for example, an acid like hydrochloric acid, the hydrogen will dissociate into a hydrogen ion and a chloride ion. And we call an acid a proton donor because they will release hydrogen ions. And a hydrogen ion has a positive charge to it. So that's why we call it a proton donor, because protons have a positive charge. So they are proton donors, and some important acids are hydrochloric acid, acetic acid, and carbonic acid that you can see there. Bases then we call our proton acceptors because they will pick up excess hydrogen ions in the solution. And when a base dissolves in a solution, it will release what we call a hydroxyl ion, which is OH negative for the chemical formula. And some important bases in the body are bicarbonate ion and ammonia. So we call a base a proton acceptor because it will combine with hydrogen ions, which has a positive charge, so it accepts those protons. The acid-base concentration is kind of measured in our pH scale. And the pH scale, this might be a test question, it's a measurement of the concentration of hydrogen ions. So your pH scale measures the concentration of hydrogen in a solution. The more hydrogen ions in a solution, the more acidic it is. And pH, if you're into math, I know we have an engineer here, I won't call you out. I had you in anatomy this summer. You guys don't need to know this, but if you're into math, pH is a negative logarithm of hydrogen ions in moles per liter that ranges from 0 to 14. And the pH scale is logarithmic. So each pH unit represents a 10-fold difference. So for example, a pH of 5 solution is 10 times more acidic than a pH of 6 solution. So between each kind of number, there's a 10 times in difference. What you guys need to know, and what will be a test question, is just to know in this pH scale, it measures the concentration of hydrogen ions in a solution, so that the more hydrogen ions we have in a solution, the more acidic it will be. And if a solution is acidic, it will have a pH range of 0 to 6.99. Neutral solutions are always have a pH of 7, and I know I'm doing a terrible job circling that. So pure water with no extra ions or electrolytes is pH neutral of 7. Anything below 7 is acidic, so it will have a high hydrogen ion concentration. An alkaline is just another word for a basic solution, and basic solutions have low hydrogen concentrations, but high pH levels. So anything basic will be anything above 7 to 14. So here's a pH scale and pH values. So these are some common household ions. So at the top, we have a pH of 14. I know it's a little hard to read here. So these will be everything basic, and then neutral, I'm just going to draw a line through 7. Your blood is just slightly basic at about 7.4. Milk is slightly basic, acidic. Black coffee, wine, more acidic, lemon juice. So kind of moving down the scale, we're going to increasing acidity. So 0 to 7 is anything acidic. And again, this anything acidic has, I'm just going to draw a high hydrogen ion concentration, and it'll be more acidic. And then anything basic will have a decreased hydrogen ion concentration, because again, the pH scale measures the amount of hydrogen ions in a solution. A strong base is sodium chloride or sodium hydroxide, excuse me. Oven cleaner is a strong base. Household ammonia, household bleach, pretty much everything you should not ingest are on the extreme ends of the pH scale, very basic or very acidic, and things that are ingestible are more in the neutral range. Neutralization. So a neutralization reaction occurs when acids and bases are mixed together. So if we combine an acid and a base together, they just neutralize and kind of cancel each other out. So it causes what we call a displacement reaction, where it forms water and a salt. You guys don't really need to know that. I won't ask you questions. A buffer solution. So buffers will resist abrupt and large swings in pH. So buffers are very important in the body because they kind of keep everything happy. So if you have a really acidic whatever coming into play or really basic, a buffer kind of comes in and keeps everything happy. Acidity involves only free hydrogen in the solution, not any hydrogen bound to anions. So buffers will resist abrupt and large swings in pH. Buffers can release hydrogen ions if pH rises, and they can bind hydrogen ions if pH falls. So basically what buffers will do if it's really basic, they'll kind of release excess hydrogen ions to bring the acidity down. Or if the pH is too acidic, they will bind up excess hydrogen ions to try to bring the pH up. Your buffers convert strong acids or bases into weak ones. And this is the carbonic acid bicarbonate system, which is an important buffer system of blood. These little crazy symbols I haven't been able to get out, but what should be here is just an arrow that goes in both directions. So this is one of your body's reversible reactions. So what this buffer system does is it takes this bicarbonate, which is a weak acid, and turns it into bicarbonate ion and a proton to kind of act as a buffering system between this weak acid and weak base. This carbonic acid and bicarbonate system, this is an important system in the body that we are going to come back to. But what I want you guys to know that this system is one of your body's reversible reactions, because it kind of controls your levels of CO2, which is carbon dioxide in the body. You don't have to understand how it all works yet, but that's one of your body's reversible systems. An enzyme in the body will work within a very narrow pH range. An arterial pH of 7, so a neutral blood during a cardiopulmonary resuscitation could predict a poor outcome. Patients presenting with an arterial pH of less than 6.85 will rarely survive. So what this is just talking about is how pH plays a role in the body. pH levels will kind of affect how enzymes work in the body. If your arterial blood pH is slightly lower than it should be, such as 7, that could result in a poor outcome. And if your blood pH ever gets to be less than 6.85, then you're just done for. It could rarely survive. So your body has mechanisms to try to control the pH levels of the blood, which again, we'll kind of get to. Organic molecules, like we said, contain carbon, except carbon dioxide and carbon monoxide. Carbon is what we call electroneutral. So carbon is kind of a special atom. It can share electrons. It forms 4 covalent bonds with other elements. And carbon is our major component, and it's one of the major components in these kind of 4 main groups of organic compounds, which are carbohydrates, lipids, proteins, and nucleic acids. And these 4 major organic compounds you should know well in terms of being able to identify like an example of what they look like and being able to kind of explain what they are. Many organic compounds are polymers, which means they are just chains of similar units. So this prefix poly means many. So a polymer is just a change of a similar unit called a monomer. And a monomer means one. So we have all these monomers kind of lining up to create a polymer. And different polymers will make carbohydrates, lipids, and so forth. They're synthesized by what we call a dehydration synthesis and broken down by hydrolysis synthesis. You don't really need to know how they're made or broken down. But it's kind of interesting to see dehydration just means that we're creating something. And when we put two monomers together, we'll take out a water. So we're kind of just taking out extra atoms so that these two monomers can bind together and are linked by this covalent bond. Hydrolysis means we add in a water so we can take kind of polymer and separate that into its two monomer parts. And this is an example of dehydration synthesis of sucrose and its breakdown. So what this is showing is that we can take a glucose and a fructose, which are two monomers, and we can put them together to create a sucrose, which is a disaccharide, which is what we'll talk about. Okay, so the first group are carbohydrates. There's sugars, there's starches, they contain carbon, hydrogen, and oxygen. There's three classes of carbohydrates. We have monosaccharides, which are one sugar, because we have a little play on words. Mono means one, and saccharide means sugar. A disaccharide is two sugars because di means two, and then polysaccharides are many sugars. They are made up of monomers of monosaccharides. So these are monosaccharides. They're simple sugars. Some important monosaccharides are pentose sugars, ribose and deoxyribose will make up our DNA and our RNA. And the most important monosaccharide is probably glucose. That's a hexose sugar. Hexose means it is made up of 6 carbons, and pentose means it's made up of 5 carbons. So that's where we get that. So here's a look at monosaccharides. Again, they're just single, simple sugars. So we have a glucose by itself, a fructose, a galactose. These are pentose sugars, so they have more of a pentagon shape because they're made up of 5 carbons. Deoxyribose and ribose are what make up your DNA and RNA, respectively. These little fun diagrams, I don't know, just brings me back to my college days of learning chemistry, but this is how in chemistry, how you draw out molecules. So each kind of point is where a carbon would be. A line would be a single bond. An O is an oxygen, as H is a hydrogen, and an O is an oxygen. And then each point, again, where you don't have anything labeled would just be a carbon. You don't have to know that. I just like to digress on that a little bit. A disaccharide is a double sugar. Disaccharides are too large to pass through cell membranes. Some important disaccharides are sucrose, maltose, and lactose, and they're formed by dehydration synthesis. So basically what we do is we just put 2 monosaccharides together and we get a disaccharide, which is sucrose. So here's a look at some disaccharides. Really simply put, they look like monosaccharides, but they are formed or connected together with this covalent bond right there. So a disaccharide is just 2 simple sugars or monosaccharides put together. And then polysaccharides are just long polymers of monosaccharides. Some important polysaccharides are starch, which is the carbohydrate storage form used by plants, and glycogen, which is the carbohydrate storage form used by animals. And glycogen in animals like us is stored in our liver. They are not very soluble, which means they don't dissolve easily. And this is a look at a polysaccharide. So you kind of see this nice hexagonal type shape, because that's kind of a typical shape, the hexagon of a sugar. And you'll see all these hexagons joined together because each one is a single sugar, and we just join them all together to form a polysaccharide. And this is what carbohydrates are. They're just long chains of these single sugars. A picture like this, you know, knowing what a carbohydrate looks like, you should know. So just kind of a picture like this of kind of a long chain of those hexagon shapes will be good. Lipids then are insoluble in water. These are the main types of lipids: triglycerides, phospholipids, steroids, icosanoids. Triglycerides are called fats when solid and oils when lipid. They're composed of 3 fatty acids bonded to a glycerol molecule. And your lipids, the triglycerides, help in energy storage, insulation, and protection. So this is what the glycerol kind of head looks like, and then the fatty acid tail. And a triglyceride consists of 1 glycerol, which is this part, and then the 3 fatty acids, which are kind of 1, 2, 3, as shown there. So this is a triglyceride. Tri for 3 fatty acids, and then the glyceride or the glycerol head. All right. Triglycerides can be constructed of saturated fat or unsaturated fat. Saturated fat just means that all the carbons are connected via single bonds, and these create a linear or a straight molecule that can pack easily on top of each other, forming a solid at room temperature. So this is saturated fat. You can see that these are all single bonds. So no, the only double bond you see is going to an oxygen, which doesn't count. So this is a saturated fat. And saturated fat is unhealthy because this kind of linear line of carbons with no double bonds can stack really nicely on top of each other. Because they can stack and pack together, it's just a lot harder for your body to break it down. So that's why saturated fat is unhealthy. Unsaturated fat just means that one or more of the carbons are linked via a double bond. So the double bonds can cause a kink in the fatty acid tail, so they cannot pack closely together. A trans fat is a modified unsaturated fat that resembles a structure. It's considered unhealthy. Omega-3 fatty acids are heart healthy. So this is what unsaturated fat looks like. You kind of see this double bond between the carbons will bend the chain. And when that chain bends, all of a sudden these 3 chains of fatty acids cannot stack nicely on top of each other because you kind of have this bend that doesn't want to fit in with the other ones. So that's why unsaturated fat is seen as more healthy, because these kind of fatty acid tails cannot pack as closely together. So we show olive oil there. Phospholipids are modified triglycerides. They have a head and a tail region. The head is polar and hydrophilic. The tail is nonpolar and hydrophobic, and they will be important in cell membrane structure. So we'll go over phospholipids a lot. Not a lot, but we'll cover it a little more in the next chapter when we talk about the cell. So this is the phospholipid structure. And you can see here, this is just showing the phospholipid structure, how they make a double lipid bilayer in a cell membrane. Steroids then consist of 4 interlocking ring structures. The most important steroid is cholesterol, which is made by the liver and often found in animal products like cheese, eggs, and meat. The steroids are starting materials for synthesis of vitamin D, steroid hormones, and bile salts. And steroids also play an important role in the cell membrane structure. So this is just a really kind of, I don't want to say a famous, but you should know what a steroid looks like. And just steroids are very unique because they have this 4 interlocking hydrocarbon rings. And this forms a steroid. So, the way that just to take a step back for a second and to think about these atoms, the way that atoms come together and form different structures in space are what will give properties to steroids or cholesterols or lipids. So it's just kind of fun. Icosanoids, I'm going to skip over because I don't think you need to know these. They are some prostaglandins. They play a role in blood clots. They have an inflammatory action that are blocked by NSAIDs. And I don't even show a picture of icosanoids, so don't worry about those too much. Then proteins are extremely important. They comprise about 20 to 30% of the cell mass. Their structure is the most varied of any other molecule. Proteins can be structural proteins, which help kind of hold cells up and give them their structure. They can be enzymes, which help to increase the rate of chemical reactions, or proteins can be involved in contraction of muscles. So proteins can be very diverse. What makes up proteins are polymers of amino acids. So amino acids are the building blocks of proteins, and they're held together by peptide bonds. The shape and function of protein is due to all of their structural levels, which we'll go through. So structural proteins, an example of a structural protein is collagen, an enzyme protein. They work to kind of help catalyze reactions. Transport proteins help to move substances. So for example, this is showing hemoglobin, which is the protein in your blood, which moves oxygen. So proteins are just super fun and exciting as well. Contractile proteins like actin and myosin help to contract and shorten muscles to help you move. Communication proteins help to transmit signals. Thank you for that. I'm enabling the live words on the screen. I always forget to do that, so thanks for reminding me. Defensive proteins like antibodies just help to protect against disease as well. So those are different kind of functions of proteins. So amino acids make up proteins, and there are 20 types of amino acids that kind of will make up all of your proteins in any kind of combination. Amino acids are formed by a specific type of covalent bond called a peptide bond. Amino acids contain an amine group, an acid group, and they differ by what their R group is present. So for example, an amino acid has this general base structure with the amine group, the acid group on one end, and then what this kind of R green box is showing, the green box will change and look a little different for one of the 20 different amino acids. Amino acids are linked together by peptide bonds. Simply put, we have one amino acid joining with another amino acid to form a dipeptide, and this is what the peptide bond looks like. So peptide bonds will join amino acids together to form different proteins. There's 4 levels of protein structure that determine the shape and function of the protein, and we're going to go over these layers of protein structure. I think there is a test question about the structural levels of protein, if you just want to kind of put a mark in your notes about this. The primary structural level of proteins is just the linear sequence of the amino acids, so just the order that the amino acids come in. The secondary structure of proteins is how the primary amino acids will interact with each other. So in the secondary level of protein structure, we can form alpha helices, which are like coils of a string, or a beaded pleated sheet, which resembles like an accordion ribbon. The tertiary structure is then how the secondary structures interact. So then the alpha helices or the beta pleated sheets can kind of fold on each other. And then the quaternary structure is how 2 or more different polypeptides interact with each other. And the next couple of slides kind of take you through these different levels of protein structure. So again, the primary structure of proteins is just the sequence of amino acids, like 1, 2, 3, 4, 5 in order. These kind of linear lines of proteins will then go on to form a secondary structure. So this line of amino acids might start to curve in an alpha helix, or it might start to bend in a beta sheet, like an accordion, but we're not done yet. Proteins will then take an alpha helix or a beta sheet and then form a tertiary structure where they're kind of folded up to form a more compact globular molecule that's held together by intramolecular bonds. Tertiary structures will go on to then form a quaternary structure where 2 or more of these polypeptide chains kind of come together. And this is where we get kind of a protein then. So this quaternary structure then is what a protein would look like, but it started out as just a line of amino acids, and it came together to kind of curve around itself or fold, kind of make a connection with each other, and then 2 or more will come together. So those are proteins, and the structure of proteins is important. The shapes of these proteins can be fibrous or globular. I don't think I asked you guys too much about this. A fibrous protein is strand-like, water insoluble. Most have tertiary or quaternary structure, and they provide mechanical and tensile strength. So we'll find fibrous proteins in your keratin, elastin, collagen. The globular functional proteins, those will be more of our antibodies, hormones, molecular chaperones, and enzymes. So these proteins are kind of sensitive because they go through so much folding that they can easily become denatured. And denatured just means that a globular protein will unfold and lose its shape. And when a protein loses its shape due to any reason, it will become inactive. So proteins will no longer be able to do their job. Fibrous proteins are a little more stable. So remember, your fibrous proteins are like collagen, the ones that are a little bit stronger. But in denaturation, the active sites will become deactivated. Proteins can be denatured or unusable by a decrease in pH. So if they get thrown into a really acidic environment or the temperature increases. Protein denaturation is usually reversible if normal conditions are restored, but it can be irreversible if changes are extreme, like you cannot undo cooking an egg, for example. An enzyme also has to do with proteins in that an enzyme is a globular protein that acts as a biological catalyst. And what catalysts do is they help to regulate and increase the speed of chemical reactions. So if you think about a chemical reaction as taking ingredients to make a product, catalysts will just help speed it up. They lower the energy needed to initiate a chemical reaction so that they allow for millions of reactions per minute. So a lot of your body's chemical reactions in the body could not continue without the work of catalysts. Characteristics of enzymes, they consist of two parts. Enzymes are very specific, so they'll only react with certain substances. And their names usually end in ASE. So we're going to talk about a lot of enzymes in future moving those around, in future chapters. And if you are reading anything about in your textbook and it ends in an ASE, that's an enzyme. So for example, hydrolase and oxidase. Enzymes will help to lower the activation, which is the energy needed to initiate a chemical reaction. And there's 3 steps involved in an enzyme action. I'll kind of go over those steps in a little bit. I don't think I ask you too many specifics about enzymes, but you should know that they help to lower the activation energy required for a reaction. So if we take a look at our reactants here, which are like our ingredients trying to form a product at the end, without an enzyme, your reactants have to kind of get up a lot of energy to get over this activation energy hump. With an enzyme, what an enzyme does is it just helps to lower the energy required for those reactants to become products. So enzymes are extremely helpful, and they do that. So for example, here's an enzyme in purple. They have an active site, which is very specific for a substrate that will bind to them. We form an enzyme substrate complex. Bonds will be formed, waters will be released, and then we form a product. So enzymes are not used up in the reaction. Enzymes can be used over and over again, but they're very specific for the reaction that they will help catalyze or help go more quickly. All right, nucleic acids are the largest molecules in the body, and nucleic acid polymers are made up of monomers called nucleotides, which are composed of a nitrogen base, a pentose sugar, and a phosphate group. And the 2 major classes of nucleic acids in the body are DNA and RNA, which we will go over when we talk about the cell and protein synthesis. DNA holds all of our genes, or the genetic blueprint for the synthesis of all proteins. So what your DNA actually is it's just kind of a blueprint or a set of instructions for how all of your proteins in the body should be made. It's double-stranded, it's helical. Nucleotides in DNA contain deoxyribose sugars, so that's where the DNA comes from, a phosphate group, and one of these 4 nitrogenous bases, adenine, guanine, cytosine, and thymine. So that's DNA. The bases will always bond to each other, but in a complementary base pair. So for example, in DNA, adenine can only pair or bind with thymine, and guanine can only pair or bind with cytosine. And just keep that in mind in the back of your mind when we go into the next chapter, when we talk about protein synthesis. So here's the structure of DNA. It's made up of the phosphate group, the sugar group, and then the base, which here is an adenine. And what we're showing here is kind of a double strand of DNA and how it'll wind together. And what keeps the two strands connected are right here, this bonding between bases. So on the right side, we have another nucleotide, a phosphate, a sugar, and thymine. And here we have adenine binding with thymine because they're complementary. Here we have cytosine binding with guanine because those two are complementary. So we have these kind of two strands of DNA, they're wound together. And what keeps the strands together is how these bases will bind together. So that's DNA. It forms the genetic blueprint for all the proteins in the body. RNA then, again, we'll get to RNA more in the next chapter. It links DNA to protein synthesis. It's just slightly different. It's because it's single-stranded. So remember, DNA has two strands that are wound up. RNA is only single-stranded. It contains a ribose sugar. So that's what gives it the RNA. It's ribonucleic acid. DNA is deoxyribonucleic acid. And the bases in RNA, instead of having thymine, thymine is just replaced with uracil. There's three variations of RNA, which we will get to in the next chapter. mRNA is messenger RNA, transfer RNA, tRNA, and ribosomal RNA is rRNA. This is an animation that I'll let you guys look at on your own, just looking at the differences between DNA and RNA. There might be a question about DNA and RNA on your lecture exam as well. Knowing the differences, knowing what they look like, even maybe knowing, I think there is a picture that's really similar to this on your lecture exam. So actually labeling a picture that looks similar to this. So take a minute to study that picture. That'll be lecture exam though. All right, then ATP, I think we just have a couple more slides to get through. And I see a question in the chat. I knew that would spark a question. Yes, so it would be like a matching question because all of your lecture exam questions will be multiple choice. If it's like a content, probably some true and false, but also like a matching question if it contains like a picture. All right, so we'll finish talking a little bit about ATP. This is another important biochemical molecule, possibly one of the most important in the body, because without ATP, we would be dead quickly. Why ATP is so important is that the chemical energy released when glucose is broken down is captured by ATP. So think of glucose as anything that you eat in the body, and glucose is changed into ATP. ATP, adenosine triphosphate, directly powers all of the chemical reactions in your cells. It offers immediate usable energy needed by your body cells. And this is the structure of ATP, which you should be familiar with as well. You can see here that ATP stands for adenosine triphosphate. So we've kind of bracketed out this whole structure that you see is adenosine triphosphate because of the adenine and the ribose make up what we call adenosine. So I don't know if you can see my cursor there. I'm going to change to my laser. Adenosine monophosphate is made up of one phosphate group. So it kind of has the bracket around the first phosphate group. Adenosine diphosphate group is made up of two phosphate groups. And then adenosine triphosphate is made up of these three phosphate groups all held together. And why ATP, adenosine triphosphate, is so important for creating energy in the cell is that these high energy phosphate bonds, which I'm circling here, can be hydrolyzed or broken apart to release energy. In general, in chemistry, in our body, whenever you break something apart or break a bond, you will release energy when you do that. The terminal phosphate group can be transferred to other compounds that can use energy stored in the phosphate bond to do group. So many times the loss of the phosphate group will convert adenosine triphosphate, which has the three phosphate groups, to adenosine diphosphate or ADP. We could lose that second phosphate group to convert ADP to AMP, which is adenine monophosphate. So really simply put, we can break apart ATP into ADP in a phosphate group. Three examples of cellular work driven by the energy from ATP. Transport work, so this is using ATP to transport things across the cell membrane. mechanical work using ATP in contracting smooth muscles, or in chemical work, ATP phosphorylates key reactants, providing energy to drive energy absorbing chemical reactions. So I will end the recording there, and then I will answer some questions.